All right, we are going to talk about the genetic code. So the genetic code is going to be talking a lot about DNA. And DNA, obviously, you know, the genes that code for everything that we are located on our DNA. And that DNA molecule also has information that contains information for how to make proteins. So what are proteins? Proteins are things within our body that allow our cells in our body to do specific jobs. So proteins do things like break things down, they perform reactions, they signal to other cells or proteins. If you remember when we were talking about the blood cells, proteins are the things that identify our blood cells as ours or our cells as ours. They help things travel in and out of the cell. If you remember the membrane structure, proteins made up those membrane channels that allowed things to move in and out of the cell. And they do a ton more things. They are very diverse. Also understand that each protein does a specific job. So when a section of DNA codes for a specific protein, that specific protein literally does a very specific job. So for example, if it's helping water travel in and out of the cell, then that protein will only help water. It won't help oxygen, it won't help carbon dioxide, it only helps water. So they're very specific in what they do for the cell. The order of the nitrogen bases is The order of the nitrogen bases, guanine, cytosine, adenine, and thymine, are very specific. So if you remember when we made our DNA molecule, I didn't tell you what order to put them in. You guys put them in random order. But in our DNA, or on our DNA, those bases actually tell a story. It's like words on a page in a book. They actually tell a story. Their order is important. Okay, and along with the gene forms a genetic code that specifies what type of protein will be produced. So for example, there's this protein called hemoglobin in our blood, and that's what carries the oxygen from our lungs into all of our cells. Hemoglobin is a protein. Hemoglobin is made from DNA, and it has a very specific structure, and its structure is made from the very specific order of bases on DNA in a specific location. And these proteins, the genetic code that tells a protein what it's going to be, is also going to tell it what its job will be. So that's kind of why DNA is so important, because these proteins do so many things in our body. RNA. So this is something you've probably heard of. I think I've mentioned a couple of times. But RNA is called ribonucleic acid, unlike DNA, which is deoxyribonucleic acid. RNA is the genetic messenger. It carries the genetic code from the DNA inside of the nucleus into the cytoplasm. And there are two types. So I want to remind you that DNA is only located in the nucleus, except during cell divisions. Okay, and when it's in cell divisions, it's wrapped up tightly into chromosomes. If DNA went out in the cell normally, without being wrapped up into chromosomes, it could be degraded or broken down. And that would be very damaging to the DNA in that cell. We need all of our DNA. So if it escapes and ends up out of the nucleus and it gets broken down and we lose some of our DNA, the cell is no longer functional and must die. So RNA allows the message that is held on DNA to travel outside of the nucleus. Okay? There are two different types of RNA that we're going to talk about. One is messenger RNA. This copies the coded message from DNA and it carries that message out of the nucleus and into the cytoplasm where our ribosomes are. Our ribosomes, remember, are our protein factories. So basically, these are the instructions for how to make the protein from DNA, and the RNA takes that message and takes it out of the nucleus and takes it to the ribosome where the protein will be made. Transfer RNA, or tRNA, carries an amino acid. Proteins are made out of these things called amino acids, and the tRNA carries those amino acids to the ribosomes and adds them to the protein according to the instructions of the messenger RNA. So the messenger RNA actually directs the operations. The transfer RNA collects amino acids and takes them to the ribosome as they are needed. All right, so DNA and RNA, they sound very similar, right? Deoxyribonucleic acid and ribonucleic acid. But they do have different structural things. So DNA is made of two strands. Remember, we talked about it being a ladder with two sides and the rungs in the middle. Well, RNA is only one-sided. It only has one of those strands, doesn't have the other, so its bases don't pair. Okay, they don't pair. There is no ladder. It's like a half a ladder. If you take a ladder and cut it right down the rungs, that's what RNA would look like. The sugar in DNA is called deoxyribose, and the sugar in RNA is called ribose. You all remember the nitrogen bases in DNA are A, G, C, and T, or adenine, guanine, cytosine, and thymine. In RNA, they're a little bit different. Adenine's still there, guanine's still there, cytosine's still there, but instead of thymine, we have this thing called uracil. Uracil it's basically the replacement. So wherever there would have been a T, there's now a U in RNA. So sometimes when we're translating, so when we have our DNA strand and the DNA strand makes an RNA, so we have to go from translating the information from DNA to RNA, and then translating the information again from RNA to protein, mistakes can happen. And those mistakes can happen in any of these three locations. When we're copying DNA during DNA replication, during mitosis or meiosis, when we're translating the DNA to RNA in the nucleus, or when we're making proteins in the ribosomes from that RNA. Anytime mistakes happen, they're called mutations. Okay, mutations. So there are three types of mutations we're going to look at. So if this is like the DNA code, it's obviously not. There's too many letters there, but I used real words because I want you to see what effect they have on the meaning of the sentence. So if this is the first one, the red hat. Okay, so there are three different words here. Now sometimes there's a substitution, and when there's a substitution, we accidentally switch one base for another. So here, the red, and instead of the H, we switched it for a C, cat. So now that changes the meaning, okay? A substitution changes the meaning of it, which can change the type of protein that's made, or change the way we pair it up. It can change the way things fold. So a substitution is when one base is switched for another. Another mutation is a deletion. That's when one base is removed. So if you look here, the rec-at, so the D has been removed, and that has definitely changed the meaning of the phrase. That no longer has any real words except for the. So that's definitely changed it, and when that happens in DNA, that also changes it a lot because it also shortens the strand, and that's a problem. And then finally we have additions. This is when an extra base is added. So here, the, an extra A has been added here, so this is no longer a word. This is no longer a word, and now we have an extra letter at the end. The ria, daha. Okay, so again, does not really make sense. It's not real words. So these are the effects that mutations can have. When each of your genes have a very specific meaning they're trying to convey, and you have these mutations, they change that meaning. And when they change that meaning, that changes the structures of the proteins and the jobs that they can do. And sometimes in humans, not having certain proteins do specific jobs can be fatal. So it's really important that our bodies get this right. So yeah, that is some information on the genetic code. I hope that makes sense. Write down any questions you have.